Welcome to Anatomy and Physiology. This is Lecture 1 of Week 2 of our second term of the course, and we will begin the study of Chapter 12 this week, entitled The Central Nervous System. As we look at the outline for this chapter's materials, we'll consider the various aspects of the brain, first looking at the connective tissue surrounding the brain as well as surrounding the spinal cord, called the meninges. We'll look at blood supply, we'll consider the blood-brain barrier. We further consider cerebrospinal fluid, where we look at the various cavities, or more properly, we call them ventricles of the brain that hold spinal fluid, and we look at the pathway of flow of that fluid. Recall ependymal cells, one of the 4 glial cells of our central nervous system, are responsible for the production of cerebrospinal fluid, which fills our ventricles as well as moves down the central cavity of our spinal cord. Thereafter, we're going to move along to look at the 4 regions of the brain, our cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. Along the way, each of these 4 regions have various subparts, and this is going to take up considerable time in our lecture this week. We'll finish this week's materials with some conversation about consciousness and the levels of alertness, as well as drugs that impact our level of alertness. Although covered in Chapter 12, we're going to push material on the spinal cord into week 3 of the term, where we'll cover the peripheral nervous system. And this is done for no other reason than the length and breadth of material encompassing the brain. What is the nervous system? Well, recall from last chapter, we said that our nervous system is a fast-acting control system that responds to the internal changes of our body as well as responds to external environmental changes by activating the appropriate muscles and glands. Ultimately, together with the endocrine system, the nervous system is one of the great regulatory systems of our human body, helping us maintain homeostasis. And further, from last chapter's lecture, when we presented the background of the central nervous system, we said it consists of the brain and the spinal cord, and that it has 3 major responsibilities. Remember, the central nervous system is responsible for integrating different kinds of sensory information that originates in the body's periphery. Further, our central nervous system is the source of our thoughts, of our emotions, and a place to store and recall memories. And finally, it's the site of origin for most of the nerve impulses that stimulate effectors. That is, instructing muscles to contract or relax, targeting glands to secrete. I want to start this chapter with the study of the brain, and to do so, we'll look at the superficial part and move deeper. Recall last time we talked about the skeleton. We said the skeleton helped protect the brain, but in addition to the protective nature of the skull, the brain, as well as the spinal cord, is covered by layers of connective tissue that further serve in a protective manner. These connective tissue coverings are called the meninges, and we have 3 of them. From superficial to deep, the 3 layers are the dura mater, the arachnoid matter, and the pia mater. Let's go ahead and look at dura mater first. Loosely translated to tough mother, this thick, double-layered membrane is composed of a very tough connective tissue, specifically dense, irregular connective tissue. Its outer layer adheres to the inner surface of the cranial bones to create what we call a periosteal layer of the dura mater, whereas the inner layer of the dura mater is called the meningeal layer, and that's going to be responsible for covering the brain as well as part of the spinal cord. The dura mater functions to further restrict brain movement, and it's composed of 3 main septa. And what I want to do next is look at those 3 septa. The dura mater functions to further restrict brain movement, and it's composed of 3 main septa, which we're going to look at a couple of slides from now. This slide is here just to remind you of what dense, irregular connective tissue looks like under the microscope. This is just a return back to the study of tissues from Chapter 4 of our textbook during the first term of A&PII. When we consider the superficial dura mater, we find that it folds, and as it folds, it creates septa of the brain. Septa refer to partitions or dividing structures that help organize and separate the brain into different regions or compartments. Specifically, we have 3 septa: the falx cerebri, the falx cerebelli, and the tentorium cerebelli. The falx cerebri is going to be found dipping vertically, or we may say dipping sagittally, into what we call the longitudinal fissure of the brain separating left and right hemispheres. So if we're looking at this particular image that comes from the PAL site, we're going to see that our dura mater is going to dip down here, creating septa separating left and right hemispheres. You can also see this here with this lateral view of the skull where we see the dip here. The infolding of our dura mater covering the brain creates the falx cerebelli, which helps separate the two lobes of the cerebellum. The cerebellum is going to be found positioned here posteriorly, and what we're going to find is that falx cerebelli is going to follow that longitudinal line to separate the cerebellum into left hemisphere and right hemisphere. Finally, infolding of the dura mater creates what's called the tentorium cerebelli, a more horizontal dural fold over the cerebellum, and so we're going to see that positioned here. Again, this is a posterior view looking anteriorly, and we're going to see this dura fold is the tentorium cerebelli, which helps separate the cerebral hemispheres from the cerebellum. And we do see that positioned here as well. Moving deep to the dura mater, we find the arachnoid manner, loosely referred to as spider mother. This middle meningeal layer lies under the dura mater to form a loose covering over the brain surface, and it's separated from the dura mater by a narrow subdural space which contains a thin layer of serous fluid, and it has delicate web-like projections that extend into the deepest layer, the pia mater, forming what we call a subarachnoid space, which is filled with circulating cerebrospinal fluid. So right now we're looking at the middle meninges or meninx, and we have right above it a subdural space as well as right below it the subarachnoid space. Finally, we have what's called pia mater, or loosely defined as soft mother, which is a thin, delicate connective tissue and a vascular membrane, which is attached to the surface of the brain to further help cushion the brain from movement, and it follows the surface convolutions of things we call the sulci, or the shallow depressions of our brain, as well as the gyri, those elevated folds. It's often compared to shrink wrap or saran wrap in that it holds tightly to the brain's surface. And one last thing to note, the pia mater is composed of two layers as well. So we have the epipia layer, which is this more superficial layer, and then we have the intima pia, which is the deepest layer. That epipia layer contains collagen fibers, and that inner or deeper layer, the intima pia, consists of elastic and reticular fibers. We now turn to blood flow of the brain, and in doing so, I want to provide you with some general details as to the importance of a good and steady blood flow to the brain. The brain is unique in that very little glucose can be stored there. And as a result of its high metabolic rate, remember I've said that neurons have a very high metabolic rate, the body must provide the brain a constant source of glucose. In fact, although the brain only makes up about 2% of total body weight in a healthy adult, it consumes about 20% of the oxygen and glucose our body uses at rest. And so with this in mind, an interruption of blood flow, even for a minute or two, can impair neuron activity. So with this in mind, we see blood fed to the brain via what we call our internal carotids and our vertebral arteries. So we're looking in this region here, and here are some examples as well. And we see return blood from head to the heart via our internal jugular veins. We turn now to a discussion of our blood-brain barrier. The blood-brain barrier is a physiological barrier, and it governs what's allowed to cross into the extracellular fluid of the brain from our blood capillaries and what stays behind in the blood. By serving as a selectively permeable barrier, it only permits some things in, and in doing so, it protects our brain from pathogens as well as potentially harmful substances that might otherwise be found in the blood. 3 structures help contribute to make up the physiological barrier we call the blood-brain barrier. We have our specialized endothelial cells with tight junctions. We're going to look at the basement membrane of capillaries and some things that surround the basement membrane, as well as we consider astrocytes. So let's go ahead first and look at the endothelial lining of the capillaries in our brain. Now, generally, endothelial cells are specialized simple squamous cells found lining vessels. And in the case of the endothelial cells making up the lining of the capillaries of our brain, these cells lack some of the traditional transport mechanisms we might find in other endothelial cells found lining vasculature in other parts of the body. Specifically, endothelial cells making up the lining of these capillaries in the brain have what we call tight junctions. And because of this, we see very little movement of material out of the capillaries and into the extracellular space of the brain as compared to endothelial cells lining capillaries found elsewhere in the body. Further, the basement membrane of our endothelial cells have special contractile cells called pericytes present at intervals along the walls of the capillaries of our brain. These contractile cells are going to monitor the composition of blood, and by contracting or relaxing, they help regulate blood flow, as well as help regulate and limit, really, the diffusion of materials out of the blood into the extracellular space of the brain. Further, we have microglia near the basement membrane. Recall, microglia are one of the 4 neuroglial cells of the central nervous system, and specifically, they have the ability to transform themselves into phagocytizing cells so as to consume unwanted materials when bacteria or viruses or other cell debris is present. Finally, we have astrocytes, another glial cell of the central nervous system. Astrocytes are responsible for maintaining the blood-brain barrier by wrapping around blood capillaries and acting as gatekeepers, so to speak, for the movement of substances out of the capillaries, things like glucose, ions, and gases that neurons need. Now, generally, what can cross the blood-brain barrier? Well, blood gases like oxygen and carbon dioxide can cross the blood-brain barrier, as can lipid-soluble substances like alcohol and anesthetics. Although water-soluble, glucose can pass the blood-brain barrier via active transport, and we also see the movement of some ions across the blood-brain barrier. In contrast, the things that can't cross the blood-brain barrier, proteins, they're just too big in size to pass across the plasma membrane. They're just too big in size to pass through the endothelial tissue from bloodstream out into the extracellular space. Also, metabolic wastes move very slowly, and most antibiotics have a hard time crossing the blood-brain barrier, and that can pose a problem because we have to be very selective in what agents can treat infections if we develop an infection of the brain. Now, exposure to some substances or medical conditions can cause the blood-brain barrier to become temporarily more permeable. Things like microwaves, hypertension, and traumatic brain injuries, or TBIs, can also lend to a more permeable blood-brain barrier. When we discuss the blood-brain barrier, we say it has 3 major functions. First, it's going to help regulate entry of nutrients from the bloodstream and into the extracellular space of the brain. Second, it regulates the removal of waste products from the extracellular space of the brain back into the capillaries. And finally, the blood-brain barrier helps regulate gases, ions, and fluid flow into and out of the brain. This slide, taken from our textbook, shows you the difference between most blood capillaries found in the body, which we would see here, as compared to capillaries of the brain by which we see the blood-brain barrier. Specifically, we're going to see larger gaps here in our normal capillaries as compared to here in the capillaries of the brain. These involve those tight junctions. Further, we see pericytes play a greater role in brain capillaries, and so we're going to see pericytes wrap themselves around, and we certainly can find pericytes elsewhere, but our pericytes are very, very active as contractile cells around the capillaries of those found in the brain. Further, we have astrocytes that contribute toward the formation of our tight junctions, and we will see the feet of some of those astrocytes wrapping around these capillaries there. We turn our attention to the brain itself, which we'll see divided into 4 primary regions. We have the cerebrum, which includes the 2 cerebral hemispheres. We have the diencephalon, we have the brainstem, and we have the cerebellum. Now, the cerebral hemispheres are composed of 5 separate lobes, which I've listed here. We have the frontal lobe, we have the parietal lobe, we have the temporal lobe, and we have the occipital lobe. And deep in this region right here, we have the insula. The diencephalon, located in the central region of the brain, is surrounded by cerebrum, and it's composed of 3 parts: our thalamus, hypothalamus, and epithalamus, which are all going to be found and represented here in purple by our textbook. And then we have the brainstem, located inferior to the cerebrum and diencephalon, and it's composed of 3 parts as well. We're going to have the midbrain, we'll have the pons, and we'll see the medulla oblongata. Finally, we have the cerebellum, which is found here, located posteriorly, and it's a standalone structure. As we move forward in today's lecture, we'll spend a significant portion of our time studying the 4 primary regions of the brain, but I want to introduce a few terms and concepts ahead of delving into the regions themselves. Let's go ahead and look at gray matter and white matter of the brain. When we consider dissected sections of the brain, which we see here, we're going to notice some regions that look lighter in nature. We say that's white matter. And we're going to see some regions of the brain which look gray. Now, if this were a frontal section of our brain, we're going to see that we have an outer cortex that's somewhat gray in nature and inner material that's somewhat white in nature. We do have a few gray structures deep here between our 2 hemispheres, but generally we see gray matter on the outside, white matter on the inside. Now, this is going to apply just for the brain. Gray matter consists of neuron cell bodies and is also made up in part by dendrites, unmyelinated axons, potentially some axon terminals, and neuroglia. But what's really important to recognize is that gray matter looks gray because the rough ER, remember those nasal bodies we learned about last chapter? They're found in the cell bodies and impart a gray color to this region of the brain by which we have collections of neuron cell bodies. There's very little myelin in this region of the brain and the spinal cord. And gray area functionally is the area where we have the integration of information. In contrast, white matter is primarily an aggregation of the myelinated and unmyelinated axons of many neurons. The lighter color is a result of the lipid-rich myelin material surrounding axons. And there are patterns to the way gray matter and white matter present. Now, I've given you the pattern that we see with a cross-section of our cerebral hemispheres and our brain, but we're going to find that that pattern changes as we move down the brainstem into the spinal cord. In this particular image, now we've taken a cross-section of the brain stem, which is found inferior to the brain. And we're going to see here we have gray matter scattered within white matter. And so we see a difference by which when we looked at this frontal cross-section here, we saw a cortex of gray matter and deeper material that was white. So we see that swap here. We can turn to look at the spinal cord and consider a cross-section of the spinal cord. Now remember, the spinal cord is going to have a central cavity filled with cerebral spinal fluid, but then we're going to see some gray matter found deep, looking a bit like a butterfly or an H, and white matter more superficial to that. The human brain contains several fluid-filled chambers or cavities that we call ventricles. These ventricles communicate with each other. They communicate with the central canal of the spinal cord, and they communicate with the subarachnoid space. The purpose of this fluid, cerebral spinal fluid, is to help bathe the central nervous system in fluids that help deliver nutrients while also removing wastes. And the fluid acts as a shock absorber. So let's go ahead and look at the 4 ventricles of the brain. We have a pair of lateral ventricles, which we see here in these reverse C-like shapes here and here. We find those very deep in our brain between the 2 cerebral hemispheres. And we have a 3rd ventricle that we're going to find here in red, as well as then we travel inferiorly and we'll see a 4th ventricle in green. The first 2 brain ventricles are the lateral ventricles, and they occupy, again, that medial region of the 2 cerebral hemispheres to create what we like to call this reverse C-shaped chamber, or set of chambers. These chambers are positioned very deep, and we see that maybe a bit better here as we're looking at this anterior view. We're looking deep into the cerebral hemispheres, but we do see it positioned laterally here and here. These lateral ventricles are separated by a membrane called the septum pellucidum. So we would see our septum pellucidum right here, and then we're going to communicate with the 3rd ventricle via the interventricular foramen, which we would see positioned here. Next, we have the 3rd ventricle, located in the diencephalon between the right and left halves of the thalamus and the hypothalamus. And it isn't paired like the lateral ventricles were, but rather it's located right smack in the middle of the brain. The fluid filling this ventricle communicates with the lateral ventricles. Again, we said that was the interventricular foramen. And then we're going to see that it'll communicate with the 4th ventricle via what we call the cerebral aqueduct. And so we're going to see our 3rd ventricle here, or we would see it positioned here as well. The fourth ventricle is located somewhat ventral or anterior to the cerebellum in the region of the pons and the medulla of the brainstem. As mentioned in the last slide, the fourth ventricle communicates with the third via our cerebral aqueduct. So we're going to see our cerebral aqueduct. Here's our third ventricle, cerebral aqueduct, fourth ventricle right here. Now we also have some openings in our fourth ventricle called apertures. In fact, the roof of the fourth ventricle contains 3 small openings through which cerebral spinal fluid exits into the subarachnoid space. We're going to see 2 laterally positioned apertures, and if we look here, we're going to see that median aperture by which we're going to then communicate with the subarachnoid space. And we see that maybe a bit better. Here we're at the fourth ventricle and we're going to see fluid then move through the subarachnoid space. Within the subarachnoid space, fluid can circulate around the brain, around the spinal cord, the cerebrum, the cerebellum. Ultimately, the fluid in the subarachnoid space creates a protective cushion to help prevent injury to the brain, while movement of the cerebrospinal fluid allows for oxygen, for glucose, and other ions to bathe the nervous tissue of the central nervous system. Now that we've talked about the cavities of the brain, our ventricles, let's consider the fluid that fills those cavities, our cerebrospinal fluid. CSF is a clear, colorless, ultrafiltrate of blood plasma contained within the ventricles of the brain and the subarachnoid spaces of the cranium and spinal cord. CSF protects the brain and spinal cord against chemical and physical injuries, while at the same time, it carries oxygen, glucose, other chemicals from the blood to neurons and neuroglia. It continuously circulates through the cavities of the brain and spinal cord, as well as around the brain and spinal cord in the subarachnoid space between the arachnoid matter and the pia mater. In total, we see about 80 to 150 milliliters of CSF in adults at any given time as a result of that filtration process by which ependymal cells are making cerebrospinal fluid. And we see a total per day of about 500 milliliters. And this gives you an idea that we need to find a way to get rid of some of this fluid. If we only hold 80 to 150 milliliters, we make about 500 milliliters. We're going to find in a future slide how we rid ourselves of that additional cerebrospinal fluid so that pressure doesn't build up within the cranium. We're going to find concentration differences in ions and proteins between our cerebrospinal fluid and blood plasma, so that's going to come into play. And finally, we're going to see that cerebrospinal fluid serves 3 primary functions. First, it aids in protection against mechanical things, essentially floating the brain and softening the impact the brain may otherwise have with the bony walls of our skull. Further, CSF aids in chemical protection. Our cerebrospinal fluid maintains an optimal ionic concentration, allowing for the propagation of nerve impulses. And when compared with plasma, CSF has a higher concentration of sodium, of chloride, and magnesium, but lower concentrations of ions like potassium and calcium. Finally, cerebrospinal fluid is circulated, thus providing the ability for nutrients to be delivered and waste products to be removed. More than 80% of our cerebrospinal fluid produced is via a network of modified ependymal cells surrounding blood capillaries we call the choroid plexus. So we're looking at the choroid plexus here, and here's a capillary, and here we see our ciliated ependymal cells surrounding this capillary and going to permit some things out of blood plasma while keeping other things in. In fact, the ependymal cells act as a sieve, resulting in the filtration of blood plasma. Something somewhat fascinating, maybe slightly off topic, but studies published in Science magazine in October 2009 by Boston University have shown that there is a significant jump in CSF production at certain times of day, specifically at night as compared to waking hours. And scientists don't yet have a full explanation of this, but it's hypothesized that this could be a result of reduced brain activity requiring less blood in the brain, whereby CSF helps replace the fluid lost to keep the brain well cushioned during sleep. Other studies back in the early 2010s at University of Rochester hypothesized that an increase in nocturnal CSF production was the result of the body's need to rid the brain tissue of metabolic wastes. So there are some various ideas as to how CSF is produced, We see it produced with the choroid plexus, but even at different times of the day, we may see more or less produced. Again, we're focused on that approximate 500 milliliters a day. And a few slides from now, we're going to see how we rid ourselves of excess CSF such that pressure within the brain doesn't increase. Let's go ahead and look at the individual steps of transport for cerebrospinal fluid, beginning at our lateral ventricles. And so this is going to be a 4-step process. Step 1 here is going to show the cerebrospinal fluid being formed at the choroid plexus. And so we have choroid plexus here in our lateral ventricles and our 3rd ventricle, as well as our 4th ventricle. So this is step 1. The choroid plexus produces CSF within each of our 4 ventricles. Next, in step 2, cerebrospinal fluid moves from our lateral ventricles here through our interventricular foramen into the 3rd ventricle. More cerebrospinal fluid is being produced here at the roof of the 3rd ventricle, and this fluid continues on its journey through the cerebral aqueduct here, which passes through the midbrain and into our 4th ventricle, and then along to the subarachnoid space, which we're going to find in this lighter blue that wraps around and surrounds our central nervous system. In the last slide, we'd seen the 4th ventricle contribute more CSF with fluid moving through the 4th ventricle into the subarachnoid space through openings in the roof of the 4th ventricle. Remember, those were called our apertures, and there were 3 of them, the median aperture as well as the paired set of lateral apertures. In step 3, now here, we see cerebrospinal fluid circulating through the subarachnoid space around the surface of the brain and down the central canal of the spinal cord. So we're seeing that movement with the help of ciliated ependymal cells along our subarachnoid space. Finally, in step 4, we need to remove cerebrospinal fluid. So of that 500 mils that we produce a day, if we focus on these components right here, we're going to see that fluid leaves the subarachnoid space via what we call arachnoid granulations, which we see here and here, and there may be others that we don't see in this particular illustration. That cerebrospinal fluid is then going to be dumped into this more superficial dark blue space that we call the superior sagittal sinus, which the fluid then ultimately makes its way back into circulation via our internal jugular veins. Now we're going to turn to the first of the 4 regions of the brain by looking at our cerebrum, or specifically we're going to look at the 2 cerebral hemispheres of the cerebrum. On first pass, this part of the brain will seem pretty straightforward, but we're going to come back later and look at it in much more depth to consider its surface intricacies. The cerebrum, commonly referred to as our intelligence center, provides us the ability to speak, to read, to write, and do all sorts of other things. It helps us make complicated math calculations, as well as helps us plan for the future, remember the past, imagine things that haven't existed before. We'll begin our study of the cerebrum by looking at some gross anatomical surface markings. First, we're going to have what are called gyri, the folds or convolutions that stick up from the brain. So we would have one right here, or right here. So these are the ridges that we're going to find. These are called gyri. We also have sulci, or the shallow grooves or furrows surrounding gyri that project downward. So if we just follow one here, or one here, or one here, these are all the shallow grooves. And now we're going to look at this deeper groove in a moment. Further, we have fissures. These are the deepest grooves between folds that help us identify different parts of the brain. In this particular image here, we have what's called the longitudinal fissure, and this fissure is going to separate the right hemisphere from the left hemisphere. And we can also consider what we call the transverse fissure. So if we're looking at our transverse fissure, we're looking right here. And that's going to help separate the cerebrum from the cerebellum. When we look at the cerebrum, we can see that some of the sulci help divide the cerebrum into several different lobes. In fact, we have 5 of them. Our textbook uses specific color for each lobe. So we're going to see we have a frontal lobe here in red. We have our parietal lobe here in this darker teal color. We have our occipital lobe posteriorly in this lighter blue, and then we're going to have our temporal lobe in this royal blue. We also, if we were to reflect back part of our frontal lobe and our temporal lobe, we would see deep to that the insula, which is indicated in that beige color. Now that we know the location of our 5 lobes of the brain, we can look at additional surface anatomy to help us differentiate these major lobes. And in fact, we have 3 sulci. We're going to see our central sulcus right here. This one's, I'd say, most critical in terms of your understanding. We're going to see then, associated with the central sulcus, we see a precentral gyrus. So pre meaning before the central sulcus, so we're going to see this ridge here. We will see then a post-central gyrus, post meaning after the central, and we see that ridge right here. So those are some landmarks we use to help identify the different lobes of the brain. We also have what's called a parieto-occipital sulcus. So imagine our parietal lobe and our occipital lobe, and we'll see a sulcus found here, which is the groove that helps differentiate the occipital lobe from the parietal lobe. And finally, we have what's called a lateral sulcus. And so we see that lateral sulcus positioned here, which helps separate the temporal lobe from the frontal and parietal lobes. And the lateral sulcus is going to run this way in more of a transverse type plane. As we consider the cerebrum, we're going to look at each of the 2 cerebral hemispheres. And in doing so, there are 3 regions associated with each cerebral hemisphere. We have what's called the cerebral cortex, we have white matter, and we have basal nuclei. So 3 regions making up each cerebral hemisphere. The first part of the cerebrum, that of the cerebral cortex, is composed of gray matter very superficially. It makes up a shell. We call it the cortex because it's just a little bit of outer surface, about 2 to 4 millimeters thick, kind of like a crust, though it's certainly not hard in nature. The cerebral cortex is commonly referred to as the seat of our intelligence, and as our textbook specifically calls it, we say it's the executive suite. Specifically, the cerebral cortex is our conscious mind. It's responsible for our general awareness, sensory perception, voluntary motor initiation. It's responsible for communication, for some of our memory storage, as well as for our general understanding. The cerebrum is also composed of white matter. So we saw that cortex here along the surface, just a few millimeters thick, and now we're moving deep to the white matter. So this is deep to the cerebral cortex. It consists primarily of myelinated as well as some unmyelinated axons in the form of tracts. Remember from our talk last chapter, tracts are just a fancy word for a bundle of axons in the central nervous system, as compared to nerves, which are bundles of axons found in our peripheral nervous system. So as we think of tracts, we should be thinking that these are the areas where messages are being sent and received. And finally, we have basal nuclei found deep to the white matter. So we're going to find that in this region right here, this is where we're going to see our basal nuclei. And they're responsible for influencing motor function, as well as they're involved with cognitive processing like attention and planning and some of our memory. Recall, nuclei are clusters of cell bodies that are found in the central nervous system. So neuron cell bodies clustered together make up nuclei, as compared to ganglia, which are clusters of neuron cell bodies found in the peripheral nervous system. So summing this all up, cerebral hemispheres of the cerebrum are the site of our conscious mind. They make up about 40% of our brain mass, and we see 3 distinct regions making up our cerebral hemispheres. Now, before we get too deep into the studies of the cerebral cortex, I want to go ahead and look at 4 generalizations our textbook makes regarding our cerebral cortex. First, the cerebral cortex contains 3 kinds of what we call functional areas. We're going to find motor areas. We're going to find 4 of them, in fact. We're going to see that those areas are responsible for voluntary movement. And we have sensory areas that are going to be responsible for receiving and interpreting sensory impulses. And we're going to look at 8 areas that are sensory in nature of the cerebral cortex. And finally, we have association areas of the cerebral cortex, which deal with the more complex integrative functions like memory, emotions, reasoning, judgment, personality traits, and our intelligence. And there are 3 of these association areas of the cortex. We'll be looking at these 3 functional areas, our motor, sensory, and association areas, in detail as we study the cerebral cortex within these next set of slides. Now, the cerebral cortex is also said to have contralateral organization. So this is point number 2 of our generalizations. Ultimately, what contralateral organization means is that the brain is divided into right and left halves. something we call left and right hemispheres. And further, these hemispheres are connected by a fibrous band of connective tissue that we call the corpus callosum. And that corpus callosum serves as a pathway for information from one side of the brain to the other. Specifically, we find that the right side of the cerebral cortex is primarily responsible for left side activities of the body, and the left side of the cerebral cortex is primarily responsible for right side activities of the body. Now, although we say that left and right sides of the brain control the activities of right and left sides of the body, that is that contralateral concept, you'd think that each hemisphere does the exact same thing, except just focused on one side of the body. But we have hemispheric specialization, and this is the third generalization our textbook outlines. Hemispheric specialization, sometimes referred to as lateralization, is where regions of our cerebral cortex from each hemisphere carry out certain functions not seen in the other hemisphere. And we see this when we look at Wernicke's area and Broca's speech area later in lecture. For now, know that some forms of hemispheric specialization include spatial recognition, include musical abilities, and include artistic or intuitive thinking. Finally, and according directly to the textbook, no functional area of the cerebral cortex acts alone, and conscious behavior involves the entire cortex in one way or the other. Now that the 4 generalizations are documented, let's go ahead and begin the study of the cerebral cortex by looking at the 4 motor areas of our cerebral cortex. Those 4 areas include the primary motor cortex, the premotor cortex, the Broca's speech area, and something we call the frontal eye field. First, we consider the primary motor cortex. This area of the cerebral cortex is located in the precentral gyrus. So here's our central sulcus. We're looking at the precentral gyrus right here. And so we're looking precentral gyrus, the frontal lobe, where we have a large number of neurons called the pyramidal cells, whose function it is to allow us to control voluntary contractions of specific muscles or groups of muscles. We say this area allows for conscious control of very precise, skilled skeletal muscle movements. Next, we have the premotor area located anterior to the precentral gyrus in the frontal lobe. So now we're looking at this position right here. So here was our precentral gyrus. We're looking anterior to that. And the function of the premotor cortex is to help us plan movements, help us control learned or patterned behaviors, as well as repetitive behaviors. And this motor area also helps coordinate simultaneous or sequential actions. We turn to the Broca's speech area located in the frontal lobe. So now we're positioned somewhere in this region. Here's frontal lobe, close to the lateral cerebral sulcus. So here's our lateral cerebral sulcus. And so we're looking at actually this position found here with these dashed lines. For most people, about 97% of people, this area is localized in the left hemisphere only, rather than being found in both hemispheres. And it's responsible for the planning of speech as well as for the production of speech. Specifically, we're looking at direct control of skeletal muscles involved with the larynx, the pharynx, and the mouth. So we are directing muscles involved in speech as well as planning speech. Damage to this area, the Broca's speech area, such as might happen as a result of stroke, leads to non-fluent aphasia, a condition which is the inability to properly articulate or form words. Patients know what they want to say, but they cannot speak the words. Finally, our 4th motor area is called the frontal eye area, and it's located partially in and anterior to the premotor cortex and superior to the Broca's area. So now we're looking at this region right in here. So we're very close to that Broca's area we just considered, but now we're just a bit superior to that. The function of the frontal eye field is to control voluntary movement of the eyeball. We've considered the 4 motor areas now of the cerebral cortex. Let's go ahead and move on to the 8 sensory areas of the cerebral cortex. Generally, sensory areas involve the conscious awareness of sensation and arrive from the peripheral nervous system, mainly in the posterior half of the cerebral hemisphere. So now we're focused on this portion of our brain. We were kind of focused on this region before. So looking at cortex behind or posterior to the central sulcus. These 8 primary areas include, first, the primary somatosensory cortex. We also have the somatosensory association cortex, the visual areas by which there are 2 components, the auditory areas, which there are also 2 components, the vestibular cortex, the olfactory cortex, the gustatory cortex, and the visceral sensory area. So we're going to go through each of these 8 areas one by one. First, we have the primary somatosensory cortex. This area is found located on the postcentral gyrus. So here is our central sulcus. So now we're looking post-central gyrus of each parietal lobe. And these areas receive nerve impulses from joints, tendons, muscles, ligaments, telling us where our limbs are in relation to space. The job of the primary somatosensory cortex is then to localize exactly the points of the body where sensations originate. And that idea is called spatial discrimination. Next, we have the somatosensory association cortex. This sensory area is found just posterior to and receives input from the primary somatosensory area. So if we saw primary somatosensory area before, now we're looking posterior to that, and we're looking at this region here. Ultimately, this association cortex is going to aid in integrating and interpreting sensations, such as understanding what an object is by feeling its size and feeling its texture. It looks at the storage of memories of past experiences and allows you to compare current sensations with previous ones. For instance, if you walked out your back door barefoot in the dark and stepped on something slimy before, when you do it again, you remember that initial slimy step and might immediately think of a slug. Or it could be the rough feel of a cat licking your hand in the dark, because that's what cats do in the middle of the night on your bed. Even without seeing the cat, if you feel that same feeling again, you may recognize and remember it as a cat licking your hand because you felt it before. You access stored memories of past experiences to compare with current sensations. We turn now to visual areas as sensory areas of the cerebral cortex, and we're going to actually see 2 areas to mention. First, we have what's called the primary visual cortex, located at the very extreme posterior tip of our occipital lobe. So we're going to find it right here on our medial surface next to the longitudinal fissure. And its function is to receive visual information from the retinas of the eye. So we're just looking at light energy and processing that as a stimulus. In contrast, we also have the visual association area, also located on the occipital lobe, and we're going to actually see that region surrounding the primary area. And it's going to receive sensory impulses from the primary visual area, as well as the thalamus, and relate present visual experiences with past visual experiences, essential in recognizing and evaluating what's being seen. For instance, you see a person with help from the primary visual cortex, but it's only the visual association area that helps determine that that person you see is your classmate from anatomy and physiology class. So we rely on both of those areas, the primary visual area as well as visual association area, to determine and differentiate. Similar to the visual areas, we also have a pair of auditory areas. First, we have what's called the primary auditory cortex, and this sensory area is found at the superior margin of each temporal lobe. And so we're going to see superior margin right up here of each temporal lobe. Now, its function is to interpret information from the inner ear, including pitch, loudness, as well as maybe the location by which some kind of a sound originates. But we also have the auditory association area, and that's going to be positioned slightly posterior to the primary auditory cortex. So now we're looking more in this region, and its responsibility is to access stored memories of sounds to identify what a given sound is that you receive at the primary auditory cortex. Another sensory area of the cerebral cortex is our vestibular cortex, and it's located at the posterior part of the insula, so we don't actually see much of that. We would see it deep right here. And it's going to be responsible for the conscious awareness of balance and equilibrium. So when we think of vestibular, we're thinking of balance and equilibrium. Next, we have the primary olfactory cortex, and it's going to be located on the medial aspect of each of our temporal lobes. So now we're looking in this region here for our olfactory cortex. We see that in yellow. And these areas are responsible for the conscious awareness of a given odor. In this slide, we have what's called the gustatory cortex, and we're going to actually see that in the insula as well. So it's just found deep to the temporal lobes at the site of the insula, and these sensory areas focus on the perception of taste. What are we tasting? Are we tasting something sweet, sour, hot, cold, bitter, etc.? That's what we're focused on as we consider the gustatory cortex. Finally, we have the visceral sensory area located posterior to the gustatory cortex. So now we would be focused on more this region back here if we're looking at that gustatory cortex in this region from the last slide. We don't see it on this map, but the visceral sensory area is involved with the conscious perception of visceral sensations like feeling an upset stomach or feeling a full bladder. So we're focused on visceral organs and feeling what those visceral organs are sending out as sensory information. Up through this point, we've discussed areas of the cerebral cortex associated with the input of one type of sensory modality, such as the gustatory cortex receiving sensory messages associated with taste, or the primary visual cortex receiving sensory modalities from the retina. But the various regions of the cerebral cortex receive and integrate streams of information coming from a number of different sensory modalities in our body as well. This experience, receiving those streams of information, is called the multimodal experience, and what it does is it's going to integrate the information coming from different modalities and formulate more of a composite experience via higher order cognitive function. In fact, some psychologists and neurologists say that this is what makes us unique. It's what makes us different from each other, what makes you different from your classmates. Even though we may hear or see or smell or experience the exact same things, how we process that information becomes very unique to each of us. And we have three multimodal association areas. We're going to see the anterior association area, posterior association area, and the limbic association area. We'll look at each of these three in the next handful of slides. The first of our multimodal areas is called the anterior association complex, and we see this here in blue. So this is the anterior portion of our cerebrum, located in what we call the prefrontal cortex. This is the part of the brain where we see development over time. It's the part of the brain that's involved with intellect, with cognition, with recall. It's also a driver in our personality. It's involved in working memory. It's involved with reason and planning, and it's involved with judgment. And I'd like to share a little backstory on the early evidence of the role of our prefrontal cortex using a story of a man named Phineas Gage. So Gage was a foreman on a railroad construction site in the middle of the 19th century. Reports indicated he was reliable, he was well organized, he was very hardworking, but one day he was tamping in an explosive in a hole drilled into rock with this very heavy, I think it's been said about 13-pound iron bar. It was about 3.5 feet long. And the charge, that particular explosive charge, unexpectedly exploded as he was packing in the explosive material. That tamping rod, the iron bar, blew out of the hole and went through the front of Gage's head, destroying a large portion of his prefrontal cortex. Upon his physical recovery, remarkably, it only took a couple of weeks, his personality was changed. Coworkers reported that Gage just wasn't Gage anymore. He was unreliable. His actions were impulsive, with very little thought to consequences. This is one way by which scientists can determine what different parts of the brain function to do for us, is through studying injuries. Another example of understanding the prefrontal cortex comes from the study of psychiatric treatment in the early 20th century, that is, of prefrontal lobotomies. While they've certainly been replaced by drug therapy, and thankfully have been replaced by drug therapy, prefrontal lobotomies tended to result in patients' lack of ability to remember and relate things over time. Attention span and the ability to concentrate were greatly diminished, and abstract reasoning became a challenge and largely disappeared in patients with these procedures. Our next multimodal area is that of the posterior association area. And so we see that area in this region here in red. So we're seeing overlapping of our temporal lobe as well as the most posterior aspect of our parietal lobe and potentially even a hint of our occipital lobe. Now, this area links information from the primary sensory areas, and it's important in perception and language. It's an important region found in this area, this posterior association area, is called Wernicke's area. And damage to this area, and we're going to see Wernicke's in this region right here, but damage to that area found in the left hemisphere of the brain can lead to various speech and language disorders, particularly Wernicke's aphasia. And individuals with this form of aphasia can typically speak very fluently, but their speech is often nonsensical and lacks any meaning whatsoever. The third and final multimodal area is called the limbic association area, and it's located in the prefrontal cortex. So we're looking at this region here in green. And this area is part of what we call the limbic system, which receives information from virtually every other association area and can therefore relate all the stimuli of an event, including its emotional context. Emotion associated with events in our life can determine whether or how long something is remembered, and it's important for survival of all organisms. Certainly, we can imagine music and a romantic moment, but we can also imagine when we're hungry, it's a great relief to find food, and then we're more likely to remember food in that place later on when we were hunters and foragers in centuries and centuries gone by. When one narrowly escapes danger, this is another example. One is more likely to avoid those predators and those places for the future. So these are just a few examples of how the limbic system can be used to determine whether or how long something is remembered as it relates to the survival of an individual. Now, we've used the term lateralization before. Several slides back. Lateralization, loosely defined, is simply the division of labor between our cerebral hemispheres. And in fact, although most of the areas we've considered in the brain up through now, the motor and sensory areas we just discussed, are duplicated on right and left, our right and left hemispheres aren't identical. Specifically, we see some regions of the right or the left are more involved in speech production, language comprehension, and handedness. Some general information about lateralization. We see this used with a term called cerebral dominance. So if we're looking at this information right here, cerebral dominance, it's simply the tendency for one side of the brain to control particular functions like handedness or speech. In terms of handedness, about 90% of humans have left-sided dominance, which means they are right-handed. Whereas the rest of the population, about 10%, we might see right-sided dominance leading to left-handedness, or we might even see a shared pattern of right and left. As it pertains to lateralization, our left hemisphere tends to control things like language, math, logic, whereas our right hemisphere tends to control things like visual, spatial skills, intuition, emotion, and artistic talents, including musical skills. Okay, so it was many slides ago, but we were originally looking at the three regions of our cerebral hemispheres, where we looked at the cerebral cortex first. Now, after all these slides, we're going to move deeper to consider our cerebral white matter. Cerebral white matter consists of myelinated as well as some unmyelinated axons and is responsible for the communication that takes place between cerebral areas, as well as between the cerebral cortex and the lower central nervous system. The white matter consists of three types of tracts, areas where messages are being sent and received. Remember, tracts are bundles of axons in the central nervous system, whereas nerves are bundles of axons in the peripheral nervous system. So we're looking at areas where messages are being sent and received within the central nervous system. We have three tracts: the association tracts, the commissural tracts, and our projection tracts. Association tracts are horizontal bundles of axons responsible for conducting impulses between different areas of the same cerebral hemisphere. And so if we look at this image taken from the textbook, we focus on these pink structures here, here, and here. What we're doing is we're seeing communication within the same cerebral hemisphere. In contrast, we have commissural fibers that are bundles of axons responsible for conducting nerve impulses between the two hemispheres. So we're going from right to left or left to right. And specifically, we're looking at this structure right here in green where we're seeing communication between the two hemispheres. Finally, we have vertical bundles of axons that we call projection tracts, which are responsible for connecting the hemispheres to the lower parts of our central nervous system, like the thalamus, the brainstem, the spinal cord, or from lower parts of the central nervous system to the cerebral hemispheres. And we see those projecting here in purple. Similar to the last illustration, this particular illustration provides additional details as to the connections we find within, between, or up and down within our central nervous system. So we see our association fibers in pink, we see our commissural fibers, which would be helping connect this hemisphere with the one that would project at you out in the screen, as well as we see our projection fibers here, by which we're going to see information between cerebral hemispheres to the diencephalon, or we may even see cerebral hemispheres connecting even further down to brainstem or spinal cord. The third basic region of the cerebrum is that of our basal nuclei. Recall, nuclei are just clusters of cell bodies of neurons found in the central nervous system. These clusters are found deep within each of our two cerebral hemispheres, and we're going to find what we call nuclei found deep within each of our two cerebral hemispheres. We find 3 nuclei, termed basal nuclei, involved in motor function, as well as involved in the cognitive processing such as attention, memory, and planning. The 3 nuclei are called the caudate nucleus, the putamen, and the globus pallidus. Sometimes you might find the first 2, the caudate nucleus and the putamen, are referred to or lumped together to be called the striatum. Basal nuclei have several functions. First, they're responsible for the fine-tuning of muscle movement while inhibiting unwanted muscular movement. This region is also responsible for ensuring proper body posture. Ultimately, these areas of the brain tend to exert an inhibitory influence on somatic motor neurons controlling skeletal muscles. So if there's some kind of a lesion or an injury or a disease in this area of the brain, basal nuclei won't or wouldn't exert the expected inhibitory action, with the result being some kind of overexcitation of somatic motor neurons activating skeletal muscles, which can lead to some kind of uncontrolled movement. Basal nuclei are also responsible for something we call associative learning. That is, we see behavior is linked with a certain response like reward or reinforcement. And finally, basal nuclei are in part responsible for our emotions, imagining that visual beauty and attraction. And so when we talk about basal nuclei, there are 2 disorders that we will look at in our lab activity. We're going to see Huntington's disease as well as Parkinson's disease. So you'll want to consider how the basal nuclei are involved in both of those diseases, one of which is a genetic disease, the other is somewhat unexplained. In addition to that, we want to look at the symptoms of each of those 2 disorders to determine what basal nuclei do and see if that's what we're looking at in terms of symptoms of these disorders. This is a good time to finish up today's lecture. When we come back, we're going to move on to another region of the brain we call the diencephalon. We'll look at the brainstem. We'll consider the cerebellum, as well as a few other components of the central nervous system before we finish up the week and prepare for the next week in chapter 13. So if you have questions, don't hesitate to reach out. Meanwhile, as always, make it a great day.